Hello and welcome dear viewers to the topic on impurity qualification, and the topic is impurity qualification based on the metabolite data. So in this topic we will see how the impurity qualification is done by various means and how the metabolite data supports the impurity qualification. So first of all we need to understand the basics of threshold for impurity levels. The three thresholds are there, that is reporting threshold, identification threshold, and then qualification threshold. So based on these three thresholds, the impurity limits are given, and if the impurity crosses the threshold of qualification, then the impurity needs to be qualified, and qualification of the impurity is required if the level of impurity crosses the threshold limit for qualification. It is, in the other simple meaning, we can say if the impurity is coming into the drug product or drug substance above the qualification level or qualification threshold, then the biological safety of the impurity is required to be evaluated for giving the specific level. So the qualification is the process of providing biological safety of the impurity at the specified level. Now we will see what are the ways are there for the qualification of the impurities. So as per the guidelines of USFDA, EMEA and ICH, some ways are there for impurity qualification, and one of them is comparative analysis, in which comparator product or RLD is analyzed and its impurities are studied. But in the current scenario, generally the regulatory authorities are not so much interested in qualifying the impurities based on the comparative analysis of the innovator product, and the impurity qualification is seen as case by case basis, and it is very product specific, as the different product will have different impurities, their degradation pathways, different compositions, different APIs, route of synthesis, and many factors are there which affects the impurities. Then another is based on the scientific literature. Then metabolite. So if significant metabolite is there as an impurity, then some principles are given for that qualification based on the metabolite data. Then structure activity relationships can be provided, and then the toxicity studies. So based on the toxicity studies also, the impurities can be qualified. So the drug metabolism is a very must to be understood topic when we consider the impurity qualification by metabolite or metabolite data. So drug pharmacokinetics involves ADME, that is absorption, distribution, metabolism and excretion, and some of the drugs may go under metabolism and may lead to the degradation of the drug and thus form the metabolite. So these metabolites may be active or inactive, and the metabolism pathways are phase 1 and phase 2. In phase 1, oxidation, reduction, hydrolysis, these are the mechanisms, and in phase 2, conjugation are the important phase or important pathway. So by conjugation, by glucuronidation, acylation, these are the phase 2 pathways. Metabolite is a compound derived from the parent drug through phase 1 or phase 2 metabolic pathways. Metabolite of the drug may be active or inactive, and it may produce the therapeutic effects, or it may have the harmful effects. Sometimes the metabolite may not have any effect in the body. Active metabolites that are those which are having the pharmacological activity at the target receptor. The activity may be greater than, equal to, or less than that of the parent drug. Many of the prodrugs are available and these get metabolized to form the active drug into the body. The example is tenofovir alafenamide and it is a prodrug of the tenofovir. Now the metabolite types. What are the types of the metabolite? Disproportionate drug metabolite is a metabolite present only in humans or present at higher plasma concentration in humans than the animals used in the non-clinical studies. So always there is a comparison between the presence of metabolite in the animal species and in the human beings. In general, these metabolites are of interest if they account for a plasma levels greater than 10% of total drug related exposure. So if you consider the AUC percentage, then if the metabolite has 10%, then you can say that it is a significant metabolite. Then significant metabolite, as per the ICH M3R2 guideline, it defines that significant human metabolites are those that occur at exposures greater than 10% of the total drug related exposure. So this is the guideline related to this definition. So generally remember that 10% is the criteria for metabolite to be a significant metabolite. Now after getting the information on drug metabolism and metabolite pathways, there are degradation pathways for the drug. Now drug degradation pathways also involve hydrolysis, oxidation, photolysis and the similar mechanisms compared to the metabolism pathways to form the metabolites. So the drug impurity formed during the drug synthesis due to degradation during the manufacturing process, stability storage or retest period may be similar to the metabolite formed in the body. And this is very much important to understand that these degradation product may be similar to the metabolites which are formed into the human beings or animal bodies. So any impurity which is also a metabolite. Now see, any impurity which is also a metabolite can be considered to be qualified. Drug product tested in clinical studies as per ICH Q3A, Q3B and some FDA guidelines also states that if the impurity is also a metabolite, then it can be considered to be qualified. And this is why, because the metabolite is present into the formulation as a impurity and also it is getting formed after the administration of formulation. So in the clinical studies, if the metabolite is the impurity, it is considered to be qualified. Common pathways for drug degradation and metabolite formation may involve mainly hydrolysis and oxidation. So if the degradation pathway and metabolism pathway are similar and may lead to the same metabolite or impurity, then the data can support to qualify the impurity as a metabolite. So qualification is based on the metabolite data and it involves the use of publicly available information for the drug substance and drug product, that is reference literature, reference product literature, clinical studies literature, API DMF, impurity literature, then literature for the animal and human metabolites, metabolite in plasma and urine. Then we have to find out the known metabolites from literature and confirm their level. Then also we have to confirm the metabolite impurity level into the drug substance and the drug product. Generally, we consider there are no clear thumb rules for topic of the qualification based on metabolite data and these may vary case by case basis. Generally, limit of metabolite impurity in drug substance and drug product cannot be given more than the actual exposure of the of that metabolite after administration in the body, and it is a little bit tedious process, and it requires the understanding of the chemistry, analysis, stability, and also the pharmacokinetics. Then it also takes into consideration the quantitative and structural estimation method which is required for metabolite and the impurities. General considerations involves the confirmation of the metabolite that it is an impurity. That means confirm that the metabolite and the impurities are similar. Confirm the level of metabolite observed in the clinical studies. Also observe the metabolites in the preclinical studies and in animal studies. Confirm the level in formulation and exposure to the metabolite in vivo by metabolic pathways in the animals and the humans. Then confirm the toxicity or mutagenicity of the metabolite. If the metabolite is toxic or mutagenic, then the qualification cannot be based on the metabolite data, and it is taken on case by case basis. The case is considered differently based on the toxicity of the formulation, toxicity of the drug, duration of the use, maximum daily dose, and the level which is observed into the formulation in stability studies. So when we arrive at the limits, basic terminologies we should understand. These terminologies are very basic, and it involves NOAEL, then HED, then MTC and MOC. So if you understand these terminologies, then the next part of this presentation will be very useful to you. So NOAEL is no observed adverse effect level. So this is the level at which the adverse effect is not observed. Then HED is the human equivalent dose. See, human equivalent dose is calculated based on the factors based on body surface area of the animal and the human, and also the animal body weight and the human body weight. Then MTC is the maximum theoretical concentration of the metabolite or impurity into the plasma, and MOC is maximum observed concentration. So MTC is theoretical concentration and MOC is the observed concentration. So NOAEL, HED, MTC and MOC are very important terminologies for the impurity qualification based on the metabolite data. See, now we will see an example considering a impurity D. Impurity D is a degradant and a metabolite also, and now you have a formulation which is having impurity D, and impurity D is a known degradant and it is also a metabolite. Then check the qualification level as per the MDD of 200 mg. If I consider a drug product or formulation, for example, we can take as a tablet of 200 mg once a day. So MDD is 200 mg. Qualification threshold as per the ICH guidelines will be 0.2% or 3 mg. TDI, whichever is lower. So it will be 0.2% as a qualification limit as it is lower, and in mg it will be 0.4 mg. 0.2% of 200 mg will become 0.4 mg. The observed level of impurity in the formulation is 0.7% to 0.8% as a degradant impurity D. Now this impurity is observed into the formulation in 0.7 to 0.8%, and thus it is crossing the qualification level, and now its qualification is required. So it will require your 100% attention so that you can understand the terminologies and understand the topic. So then get the complete pharmacokinetics and pharmacodynamics information of the formulation, that drug, then drug pharmacokinetics into animals and humans with dose and duration of the treatment or duration of administration, maximum duration of administration in any disease condition, and then understand the in toxicity of the metabolite from the literature, and then get the NOAEL, that is no observed adverse effect level, which is given in mg per kg and mg per kg per day, that is mg per kg per day. So NOAEL is no observed adverse effect level in mg per kg per day for the animal. Now convert the NOAEL to HED, that is human equivalent dose, and consider 60 kg as a normal adult human body weight. HED will come from the calculation that animal dose in mg per kg into animal weight in kg divided by the human weight in kg. So conversion of animal doses to human equivalent doses is based on the body surface area and it requires a factor. So in the guidelines you will get these factors which are specific for the animals. I have considered a factor of 6.2 for rat to human dose calculation. See, the studies are performed in the animals and then from there the data is taken for the human trials. And these dose calculations require these factors which are based on the surface area and the body weight. Now we have NOAEL factor and the body weight. HED will be, if I consider a NOAEL level of 0.25 mg per kg per body weight, 6.2 is the factor and 60 kg is the body weight. Now this calculation will give 2.42 mg as a daily dose or daily exposure of the impurity or daily exposure of the metabolite. So 0.25 mg per kg was a NOAEL in animals, and now we have 2.42 mg per day as a limit qualified for metabolite or impurity based on this HED calculation. So can we take this 2.42 mg as a qualified level? So some factors are also there. We will try to understand in detail whether we can take that limit or not. So calculation given us the 2.42 mg per day and as a qualified limit. The MDD is 200 mg, then for 200 mg 2.42 mg is the limit, and if you convert this limit of 2.42 mg into percentage, it will be 1.21%. Now check the impurity level observed in the stability studies and then confirm the limits. So deriving the specifications. Now you have MDD. The qualification threshold is 0.2%, which is 0.4 mg. The observed level in final prototype in the stability study is 0.8%. This is considered as a maximum level of impurity D, and if 0.8% is converted into mg, it is giving 1.6 mg. Now the limit as per the metabolite data from literature and the clinical and preclinical studies in animals, we got calculation of 2.42 mg and a limit of 1.21%, considering the 200 mg maximum daily dose. So now can we give a limit of 1%, as 1% will give a 2 mg limit, and as per the calculation we have limit of 2.42 mg. So let's see whether we can give it or not. Now there are two terminologies like MTC and MOC. So MTC is maximum theoretical concentration and MOC is maximum observed concentration. And this MTC and MOC are for the impurity or metabolite. Now check the bioavailability of the drug and the metabolite. So there will be two cases. The bioavailability will be 100% or less than 100%. So two cases I have considered here. The bioavailability of 100% will give the MOC, that is maximum observed concentration of 2.42 mg in the body. And at this case 1, the MOC and MTC will be equal. Now case 2, if I consider a 50% bioavailability, then 2.42 mg in the body will become 1.21 mg in the body. And now the maximum observed concentration is less than the maximum theoretical concentration. Now see here, the MOC is less than the MTC. Consider the actual specification of 1%. It is 2 mg in the body. And now if you have case 2 of 50% bioavailability, then can you give the specification of 1%? So this is very important to understand whether we are lying now. The case 1 or case 2. Now see, the observed is greater than or equal to theoretical, then it is qualified. So if your observed level is greater than or equal to the theoretical level, then it is qualified. And now you can give the limit of 1% or slightly above the 1%. If the observed level is less than the theoretical level, then it is not supporting the qualification based on the metabolite data. And for this type of situation, further studies are required. So this situation is considered for case by case basis. For this, data is required to prove that the impurity is metabolite or the metabolite and the impurities are same. Then PK studies and data for urinary excretion, absolute level in the body for that metabolite or impurity is required. Data for exposure across the species is also required. Sometimes the animals will have more metabolite circulating into the plasma, but when it is given to human beings, the story may get changed. The metabolite may be higher or metabolite may be lower than the calculated limit. So once again it is required to be proved that the impurity is metabolite. PK data is required to be given and data for exposure is required to support the qualification. Consider the bioavailability or plasma concentration of impurity and confirm the limits. In the present example of 100% bioavailability, the limit of 1% can be given because the maximum theoretical concentration and the maximum observed concentration, if it is compared, then 1% limit can be given as the maximum observed concentration is less than the theoretical concentration. So mainly the requirement for qualifying the impurity on the basis of metabolite requires complete understanding of the pharmacokinetics of the impurity. Then sometimes metabolites in safety testing is required. Case by case basis evaluation is done for the qualification of impurity on the basis of the metabolite data. See, there are various examples of drugs and formulations for which the impurities are qualified based on the metabolite data. and some of the examples are oxycodone, brigatinib, levofloxacin, deslansoprazole, lacosamide, vildagliptin, and enzalutamide. So this is regarding the qualification based on metabolite data. This is part 1, and I have covered basic terminologies and basics of the impurity qualification based on metabolite data. And in part 2, I will try to cover the exact calculations and the case studies so that the viewers will understand the qualification process. See, impurity qualification is very much important for safety and efficacy, and also to protect the public health. And impurity qualification is also a very favorite topic of the interviewers. I hope you might have got a good understanding out of these videos. Keep watching the videos and please do like, share and subscribe to Pharma Learning in Depth channel. Thank you.